Our keynote speaker for today, Associate Professor Siouxsie Wiles from the University of Auckland in Aotearoa, New Zealand. Siouxsie describes herself as a microbiologist and a bioluminescence enthusiast, but to many, she is that pink-haired science lady. She studied medical microbiology at the University of Edinburgh, followed by a PhD in microbiology at the Centre for Ecology and Hydrology in Oxford. She spent almost a decade at Imperial College before relocating to Aotearoa, New Zealand as a Health Research Council Hercus Fellow in 2009. She currently heads up the Bioluminescent Superbugs Lab, where she combines her twin passions to understand infectious disease and to identify new antibiotics. She has a keen interest in demystifying science. She's an active tweeter, blogger, podcaster, and media science commentator, and has worked with artists to make living works of art for various exhibitions in Aotearoa and overseas. In 2017, she published her first book, Antibiotic Resistance, The End of Modern Medicine, ? And in 2019, collaborated with her daughter to make a children's show about microbiology. Siouxsie's won numerous awards for her research and science communication efforts, including the UK National Centre for Replacement, Refinement and Reduction of Animals in Research, 3 Hours Prize, the Prime Minister's Science Media Communication Prize, and the Royal Society Te Apārangi Callaghan Medal. In 2019, she was appointed member of the New Zealand Order of Merit for services to microbiology and science communication. And in 2021, she was named the Kiwibank New Zealander of the Year for her public communication work concerning New Zealand's response to the COVID-19 pandemic. It's my pleasure to ask Siouxsie to speak today. Over to you, Siouxsie.
Thank you very much. And thank you so much for the invitation to come and speak. It's actually nice to be talking about research and not COVID for a change. So let me just see if I can share my slides. Oh, can someone enable that for me, please? So what I'm going to do today is explain a little bit about what you've heard in my bio, about what my research is, explain a little bit about the importance of open access for my science communication work, but then also talk a little bit about what we're trying to do in my lab, broadening access to our research beyond just publishing in open access journals. All right. So hopefully you can see that okay.
Looks good.
Great. Okay, so yeah, so what I really want to talk about is open access and open science from a scientist's perspective. So as you've heard, I'm a microbiologist and I am absolutely obsessed, I guess I would say, about bioluminescence. So this is the production of light from living creatures. And here we have a picture of a bacteria that glows in the dark. This is a completely harmless natural bacteria that lives in the ocean. And basically, when you look at it in the light, it just looks like a kind of culture of bacteria. But if you turn the lights out, it makes this really incredible blue light. So this is essentially a chemical reaction that lots of different creatures use and essentially makes light as a byproduct of this chemical reaction. So in my lab, we're really interested in using that chemical reaction for all sorts of different things. One of the really important things about it is that we can use it as a surrogate for looking at bacterial numbers. So essentially, it's like the lazy microbiologist way to do microbiology. Instead of counting how many bacteria there are in our cultures, just by plating them out onto petri dishes and waiting for the colonies to grow, we can actually just measure light by putting the samples in a luminometer. And so what you can see here on this graph is essentially the more bacteria there are, the brighter the light will be. So we can use that light as a measure for how many there are. But it's also a measure of whether they're dead or alive, because only living bacteria will glow. So it essentially gives us a really quick and non-destructive way of looking at how many bacteria we have, where are they dead or alive, in minutes rather than the days or weeks or months it can take different species of bacteria to grow on petri dishes. So in my lab, we're applying this in lots of different ways. One of the ways we are applying it is to look for new antibiotics. So these are essentially compounds that kill bacteria. And one of the places we're looking is from New Zealand fungi. So we have a collaboration with Manaaki Whenua, who are one of our Crown Research Institutes here. And essentially they have a large collection of fungi that have been collected over about the last 60 years. There's something like 11 or 12,000 different isolates in their collection. And we are basically growing them to see whether they are able to kill our bacteria. One of the reasons that we're really interested in looking at fungi is because one of the first antibiotics ever developed, penicillin, comes from a fungus. And actually, fungi were sort of initially an interesting source of discovery for antibiotics, but then soon were displaced by bacterial species. So we have this collection here that's never really been properly examined. And so we're wondering whether there might be some novel compounds that might make new drugs in there. So the idea is that our collaborator, Bevan Weir, who's the essentially the custodian of this or curator of this collection, he gets the fungi out of the freezer for us. We then pit them against our glowing bacteria in my lab. So we're looking for things that will basically reduce the light. And then if we find something, we grow it up, the fungi that is, in large quantities, and then send it off to our chemists to try and find out what's in there. The idea being that we hopefully don't just discover penicillin over and over again. I can honestly say we have not done that. So here's just one of the ways that we do that. This is actually our kind of old-fashioned way where we grow the fungi on petri dishes, punch out little holes, and then we fill those holes with our different species of bacteria that we've engineered to glow. And that way we can just use light to say, is this fungus producing anything that can kill any of these bacteria? So we can look for things that kill all of our nasty bacteria or just some species. We've now kind of miniaturized this so that it works on what we call 24 well plates. So it's basically, we can get, instead of a stack of 24 petri dishes, we've just got it in this really tiny area. So that's one thing we're doing with it, making our way through this collection of thousands of fungi. But the other thing I'm really interested in and have been interested in for a very long time is what makes microbes infectious. So one of the ways that we have of studying this is a bacteria that causes food poisoning in mice. And one of the really cool things about light is it travels through flesh and skin. So we can use these glowing bacteria to look at where bacteria are inside of living animals. So here's a sleeping mouse that's got this dose of food poisoning bacteria in its belly. It's what we call a fecal oral pathogen. So it basically goes in the mouth and then comes out the bum. And so what you can see in the other picture is essentially the mouse cage with those contaminated poo with the bacteria all over it. And so we've used this as a way to understand how bacteria evolve as they move from host to host by essentially putting fresh animals in these cage, uninfected animals, and watching the bacteria transmit from animal to animal. We know when they're infected because they start to glow. And then we basically take the bacteria at every stage to see how have they changed over the course of that infection. So this is how we're kind of using bioluminescence in my lab to try and understand essentially how do bacteria evolve to make us sick and to hopefully try and find new medicines. So one of the things as a researcher, though, that that is just sort of fundamental to my research is that it doesn't end with a journal publication, right? So we do the work, and then normally the way we're incentivized is we write that research up and we publish it for our peers. But really, and I think you can really see this during the pandemic, right? That research may be of use, of interest to more than just our peers, right? In a pandemic, we've seen how important it's been for, for policymakers, for healthcare workers, for everybody. So more than a decade ago, I started trying to learn how to communicate better to different audiences beyond just my peers using this sort of publication in journals. So I kind of like to call myself a scientist communicator because I think this communication of our research to broad audiences is really, really important. So I've become sort of one of the go-to people to talk to the media about microbiology related stories. You know, discussing things with the media is really different to other audiences. They work on really rapid timelines. You know, you can't ring them up and say, "Oh, I've got time tomorrow." They usually want you in the next 5 minutes. But that's an audience that I kind of learned how to talk to. I've also been a science commentator for one of our radio shows, so the 9 to New program, which is on Radio New Zealand, which is sort of the equivalent to ABC, I guess, in Australia. And the idea with this slot is that basically once a fortnight, I get the opportunity to pick 2 to 3 science stories and just talk about what they are. So I've tried to not cover microbiology every fortnight. But I've also tried to talk about the process of science. You know, how do we find things out? How do scientists do different kinds of experiments? I also write a column for one of our media organizations here called Stuff that goes in some of the printed papers. And that's every fortnight again, and it's just sort of a 500 words to talk about different kinds of research. This was quite early on last year, talking about the rollout of COVID vaccines and how it wasn't equitable, and based on some research talking about how you could build a sustainable vaccine industry using synthetic biology. And then I've also, during the pandemic, started writing for a little media organization called the Spinoff, basically writing about the pandemic, talking about trying to keep people up to date with what the latest research has been. And as part of that, I've ended up collaborating with a cartoonist called Toby Morris, and we have made probably about 50 graphics now, trying to communicate different things about the pandemic or different things about the virus in sort of shareable forms that can basically be released to anybody. What we have done is release them under a Creative Commons license, because it was really important to me that if we made something that was useful, that people would be able to translate it and use it, adapt it for their own purposes. So you might have seen our very first graphic. It was to communicate the idea of flattening the curve. So this was back in March of 2020. It was really interesting to see it held up by our Prime Minister, Jacinda Ardern, in one of her press conferences. But it kind of went viral around the world, people using this kind of graphic to talk about this concept. After that, so we then this started our collaboration, and we then did a whole bunch of graphics that were around everything from how variants arise to what are the different approaches to try and deal with the pandemic. So all of this would have been impossible, though, if it wasn't for open access, because what's been really, really important to all the science communication beyond my own research has been to have access to scientific knowledge. My university subscribes to lots of journals, but not all of them. And so having access to articles that are not behind a paywall has meant that I've actually been able to talk about them or write about them for other audiences. What I really like to do is to put links to those articles in whatever it is that I'm writing about or talking about, so that people can go back and read the primary literature themselves. And I'm more likely for my columns to write about research if it is open access, because that then means that others can actually go back and see what the research is themselves, right? Do they agree with my analysis of it? Do they not? Whereas if it's behind a paywall, it's obviously difficult for people to do that. But as I say, my communication beyond my own research would have been very difficult if it wasn't for ability to access that particular research. What I want to move on and talk about, though, is how it's not just access to the peer-reviewed publications that is important. So I've been really interested in, for quite a number of years now, in how do we do science better? How do we do it? How do we make sure that we're using the precious resources of people, time, and money more appropriately to understand, the problems that we're facing and to try to come up with solutions. So one of the options is to do science more openly, and it's really interesting how when you sort of talk to people about what open science is, it's kind of like, oh, you just do exactly what you're doing, only you just like don't do it in secret, which is kind of how we do science at the moment. There's another strand, though, to this openness, and that's how do we make science more reproducible? So there's lots and lots of studies have shown that, you know, if you take a certain finding and try to reproduce it many, many times, it's not reproducible. Or the experiments themselves aren't reproducible because it's very difficult to get all the information around how things were done. So I'm kind of interested in how all of these things interplay and how, as a practicing scientist, we can, you know, incorporate these different things into my lab practice. So I just want to start by talking about what the process, I guess, of science is and where these different parts come in. So this is a very kind of linear way, I guess, of thinking about science, but or research in general, which is to, you know, you start by asking a question, or having a question, you know, you do your background research. Has somebody answered that question before? What else do we know about that topic? You know, from there we go on to construct a hypothesis, then to test that with an experiment. You know, did that work? Did it not work? If it didn't work, can you troubleshoot, find out what was wrong? Finally, you might get some data, which you can then analyze and draw conclusions on, and then essentially communicate your results. And there's this kind of feedback loop where, you know, as you get more data, that might mean you change your hypothesis. It kind of feeds back in, and then you would change your experiments. Obviously, all of this costs lots of money. And, you know, I think when we write grants, we often write it in this way that, you know, we have a hypothesis and we're going to do this stuff. What tends to happen is that people are already quite far down the track. They have a lot of data already, you know, they're writing up their stuff. And so essentially the grant they're writing is almost to help them fund their next things. So it's a sort of an interesting way where we present it as kind of this linear process, but it really doesn't kind of work like that in real life. Of course, we also have, you know, little groups all around the world doing this. So we have these processes happening all over the place with different people in different stages within these different processes. And that's really important, right? Because when I talk about how we best use you know, resources that are precious, people, time and money, there might be lots of things that are being done that are being repeated potentially, that nobody really knows about. Okay, so if we break down this process then and see where open science and open access can really help. So in the first instance, this ask a question, do background research and construct a hypothesis. Well, it's really important then, you know, to understand what's published or available, right? Has somebody asked that question before and answered it? If they've answered it, have they published it? You know, what's available? And this is where open access is really, really important, but also what we publish, right? So what really infuriates me is that we might do something, find that it's a dead end, and it turns out probably others have found that that was a dead end too. If we get to the experimental stage, you know, we're going to sort of set out a series of experiments and try and, you know, develop some protocols that will work for that. The question here again is what's published, what's available. So has somebody already tried that and it didn't work, but we don't know about that? Or has somebody you know, got a protocol that they put in their paper that you're really interested in reproducing, but can it actually be replicated? Often not, because so much of the detail is kind of missing when we actually just publish our manuscripts. When we get to say we've managed to do some experiments, we now have some data. When we get to that data and you know, drawing conclusions and communicating our results, the questions here then are we communicating things warts and all? You know, have we completely documented the process? You know, what are we communicating? And it's really clear that the way that things currently stand, you know, we don't document everything. Very, very few people document everything, and we certainly don't publish everything. It's very hard to publish everything, right? Often you have this kind of story, and you know it's the story that kind of that you manage to get published, and all of the dead ends are bits that are just often removed by reviewers. So I just want to explain a little bit about kind of, I guess, my journey to all this. So one thing that's been really important to me is, especially with this antibiotic discovery project, was showing people what we've done and publishing all of our raw data. So here's one of a paper that we published a few years ago. We've got all this data. This is around Mycobacterium tuberculosis, which is the bacteria that causes TB. And we were looking at basically developing a new way for testing TB to look for antibiotic activity. And so one of the things I wanted to do was include the data that we, that was behind our graphs, so that if others wanted to check that it was correct, they could actually do that. And so what we did was put the data up on Figshare. And this is where I learned that as a scientist who uses programs like GraphPad Prism to graph my data rather than doing things using coding, that the way that humans deal with spreadsheets is completely different to the way that automated programs deal with spreadsheets. And so we just put our data up thinking we were making it, accessible, but actually it turns out that it isn't really that accessible at all. So one of the big learnings for me was understanding what tidy data is, how you make your data machine readable, how you can put it in a format where more people will understand and be able to use that data if they wanted to. So more recently, we published another paper, this again about mycobacteria, but this was starting to be stuff from our fungi project. And what we've learned now is basically to put our whole protocols, all the stuff that doesn't end up being in an actual paper, in a place so that we can say, if you want to reproduce this, here is exactly the way that we did it, but also put our data in another place in this format that is more machine readable, more complete and understandable. So what we have done is basically put all our protocols up on a protocol repository website called protocols.io. So we have an account there, and essentially for everything that we do now, we're trying to put together a protocol that says this is exactly how we did it. It has everything in it, temperatures, all sorts of things, stuff that you would have thought would be put in a methods paper, but often is the little bits and pieces that are missing. So we now have those as standard. What's really cool about these is if you make a change, you can change it on the protocol here and then say why you have made a change. And it keeps the record of how the protocol has changed over time. It gives them, putting them up on this website also gives them all a DOI. So that's, now our protocol has a kind of a history and a format to share. One of the other things I've been really, really keen to explain to my students, I guess, is that often when you know how to do something, when you're trying to explain it to someone else, you kind of assume that they have knowledge that you have. And so you might miss steps out because they're just completely natural to you. So what I do now is I get my students to watch this really fantastic video, which is basically, it's called the Exact Instructions Challenge. This is a dad and his children. He's called Josh Darnett, and he basically has done this really cool thing where he gets his kids to write down some instructions for how to make a peanut butter and jelly sandwich, and then he does exactly what they say in the instructions. It's absolutely hilarious, but it's a really good way of getting people to go, oh, I see how actually I didn't write that bit right. So what you can see in the picture here is that the instructions were for him to basically to spread the bread with a butter knife. Obviously, his child meant you dip it in the peanut butter first, but that doesn't what it say. It doesn't say that on the instructions. So you can see here he's spreading the bread around with that butter knife. So I get them to watch this and go, what are the things that we might be missing? Really important for us is to get our protocols read by people who are not doing the same work as us. So again, they can kind of say, oh, actually, this bit's not quite clear. So after that then is putting up our actual data. And so now what we do when we're publishing a paper is we basically put our data, say in this tidy data format, we put it up on Figshare, and then we insert the link to that in the paper as well so that people can download that. And it's been really amazing actually seeing people actually do. People look at the data and people download it. It's I don't know what they're doing with it, but it's been really interesting to see people actually do that. So sort of to finish off, I guess the one thing that still frustrates me though, is that when we when we publish our work, we're really publishing this kind of, as I say, the story that goes from start to finish in a very, in a way that kind of explains, here's the things that we have found. When we I guess, think of that in terms of going through a maze, it's like you only really had one path. But when we actually look at what we do, you go down all these dead ends, you do all this stuff, none of which ever appears in the paper. So I'm really interested in how do we better capture what it is that we do so that we can report all of it. And I think this is a really, really cool little diagram of that by an artist called Red Pen, Black Pen. So we can think of the paper that we publish as kind of our house, the supplemental data, which is often the place where we put some of the kind of related things, is this kind of, think of that as the basement. But actually everything else we did is kind of like the monsters under the basement, right? So how do we show everyone what we've done with those monsters in the basement? How do we learn from that? This is something we're still grappling with. In my lab, what we're building now is a kind of a portal that we in real time put our data into. So with everything that we're doing. And so we will, we might have fungi that we never report in our publications, but they will always be here on the portal. So trying to think about where's our data? How do we report everything? We're kind of focusing on one project at the moment. What is still not really clear to me is how we do this for other projects that we have. Not that anybody might look at it, but how do we report everything that we're doing? So one of the things that I've learned is that doing open and reproducible science is really hard. I think there's when I first came into it wanting to do open science, what I actually wanted to do was reproducible science. What I want to do is be able to say, this is exactly how we did it. Here's all our stuff. And these things are not similar. They're not the same, but they're kind of related. And so it can be easy to do something openly where you just sort of show everybody your workings, but that's not necessarily actually letting people, it's not fully documenting everything. I hope that makes sense. Anyway, so this is hard. It's largely unsupported. I don't know where to go to support, for support. There's some people doing reproducible science. There's some people doing open science. It doesn't feel like there's people doing sort of both those things combined. And it's really actively disincentivized by certainly my institutions, promotions, things, everything. We're really pushed into, can you publish as many papers as possible in, preferably high impact journals, rather than actually just showing people everything that we do and making that all available to others. So I think we need changes in the way it's incentivized, and we really need support to understand how to do this better, because documenting stuff is really hard. And often it takes a lot of time. And so, I have students in the lab who they don't want to spend all their time writing down exactly what they're doing and exactly which bits they've used, what batch of, what media they used for this, which is kind of what you need for reproducible science. So it's hard and I feel unsupported. But I'm really keen to do it. So I'm open to suggestions if people have ideas of things that would help us. And with that, I'm happy to answer any questions or have a discussion about how we do this sort of thing better. So I'll stop sharing and thank you for your attention.
Thank you, Siouxsie. That was absolutely fascinating. And I'll definitely be going back to that presentation, and your timing was immaculate as well. So thank you. Please do place your questions at the Padlet link provided. And I can see that we already have a couple of questions there. So firstly, Siouxsie, do you think the format of scientific papers also needs to be reassessed to make science more open? Removing the paywall is one thing, but making science more accessible also depends on how it's communicated.
No argument from me there. I mean, I guess there's two issues, right? So I absolutely agree about so the paywall is one thing where it's really blocking access to people. At the moment, it's blocking access to people who have, who don't have the means to subscribe to that journal or whatever. And so that, locks out a huge number of scientists all around the world and researchers around the world. But you're right. I mean, my argument is also that the open access is about who else gets access to that knowledge. And the way that we present that knowledge at the moment, actually, it's getting worse and worse, right? It's getting more full of jargon. If you read Nature papers now compared to what they were 30 or 40 years ago, they're very different. They're really for experts. So we do need something else that bridges that, whether it's another version of the paper or a bit of it that kind of helps communicate it in a different way. But we have to acknowledge that that's a different skill to writing an academic paper. And it's one that takes practice. So I really like, there are funding agencies, I don't know what it's like in Australia, but certainly, the Wellcome Trust in the UK, they have requirements now where science communication is kind of part of their funding applications, right? There will be an acknowledgement that you're going to communicate this in ways beyond that academic publication. But I think there's also room for how do you put a layer on that, something associated with that publication that helps to communicate it to whether it's policymakers or healthcare workers or something like that. So, and I think, there have been some attempts at a sort of a lay summary. Again, those things take time and most scientists cannot write a lay summary, said as somebody who used to sit on an animal ethics committee and, never. It's really interesting how when you're so into a subject, you kind of don't, you lose those skills. So it has to be something that's incentivized and has to be something that's supported.
Thank you. There's a few more questions coming up. The next one, also, what led to protocols.io being chosen over other platforms such as OSF registries? That one you can answer?
Good question. It seemed a bit more intuitive to me. I mean, I'm kind of a bit platform agnostic. I think we spend a lot of time worrying about platforms. What it really needs is to be somewhere and to have that DOI. And that's kind of what that provided me. In the time that I've used it, it has changed. So it sort of was all free. Now, of course, it's gone to a sort of more subscription model. I've kind of had a play with that other one. It feels a little bit less intuitive to me as someone who doesn't really code and stuff. But as I say, I think we get a lot of, we get hooked up on the platform, and what we should be thinking about is what's the principle and how do we make sure that wherever it is accessible in the appropriate format and has a DOI.
Thank you. Some OA publishers, like Frontiers charge extortionate article processing charges. What effect do you think this has on the development of science and emerging scientists? Should OA publishing be non-profit?
Yes. Really tricky, right? I mean, there are clearly costs associated with it. There are costs associated with all publishing, and it's very clearly, none of it actually properly, how to say this? It's clearly a hugely profit-driven industry, regardless of whether you're open or not, right? So we know that Elsevier and all of those have got higher profit margins than Apple and things, right? So this is hugely profitable. And it's profitable because most of the work is done for free, right? By people who like academics and stuff. So the whole model is wrong. That I object to having to pay to make it open. But in the absence of it being free in any other way, I will pay the charges. I'm kind of in this really interesting space where I have a pledge that we will put all of our work open access. So that determines where we publish, right? I would actually quite happily just publish all of my work in PeerJ. I really like their model. They, you know, but I know that that's also not acceptable from a career development perspective for both myself and my students. What I really like about Frontiers, while they're very expensive and they certainly some of their journals have had a real kind of, you know, are embarking on some predatory behavior, I love the way they do their peer review. You know, they have this, they have essentially like a dashboardy type thing, or no, like a space where the researchers and the peer reviewers can come together and discuss it. So I really like, that's kind of why I've published there a few times, because I really like and support that model of peer review. But it's a mess. The whole thing is a mess. The whole thing should just be burnt to the ground, frankly, and start again. But it's, again, that's all completely disincentivized, right? And I'm hoping to go for promotion at some point, and I'm going to have to put a statement about why I publish in certain places, because it's nothing to do with where the work could go. It's to do with my values about how I want the work to be used and viewed.
Thank you. I think we have time for another question. My university has committed to an open research culture. This is one way we hope to make the issues you have raised visible and tangible. We hope to have a network of academic open research champions help us get the word out. But don't universities need to motivate this from above?
No argument from me. As I say, I mean, I think it needs to be, so it needs to be supported and it needs to be incentivized. And at the moment, the institution that I'm at doesn't really do either of those things. I'm not, you know, at every one of my academic performance reviews, I've talked about this for years. This is how I want to behave. This is what I want to do. What support is there for me? And my head of department can't help me with that. So we may well have things happening at the university, but I don't know how to become aware of them. I don't know who to talk to, you know, and I think this is a problem we have. So the solution, I think, at an institution level is exactly this, to say, actually, this is how we want to do it, and we're going to have champions, and that requires resourcing and support. And I'm really envious that you have that, but also really keen to hear how it goes. And I would love, whoever that person is, if you connect to me, I'd love to know, I'd love to connect to those champions to understand, you know, what they're finding out, and yeah, how we move that to other organizations.
Thank you. I think we can squeeze in one more quickly. What help, if any, have you received by libraries or librarians in navigating this space? Would you like more library support in future from libraries and librarians? And if so, what would this look like?
Oh, that's a great question. I think that, yes, I would like more support. I mean, I didn't even realize until recently that our library had negotiated some kind of article processing kind of agreement with some journals. So there's a real issue, I think, in how information is disseminated and who gets to hear it. And I think this, I mean, our university website is appalling. It's hard to find things. So knowing, yeah, how you make processes so that you know what help is available, I think is really really important. In terms of the open science stuff, see, I'm not even sure what help I need. I mean, maybe are there places where people would prefer that we put our protocols, or, you know, curating that kind of information, I guess, would also be really useful. But I think maybe just a bit like the previous questioner, having communities of practice, having people who are on this journey so they can be on that journey together, and perhaps the libraries are a resource for that, for bringing people together to help, you know, navigate this kind of area.
I'm afraid we'll have to end it there. Thank you so much, Siouxsie, for joining us today. your brilliant presentation, which we will be sharing with everyone after the event. Thank you everyone for your questions. As Siouxsie has said, she's happy to hear from you if you wish to continue this conversation. So thank you for joining us.
Thanks for having me.